you can see the log ratio here. It's a little bit equivocal as to whether there is an actual loss there or not. You can see maybe a little bit of dip in the log ratio. And so if you zoom in on this region in the genome and start looking at the BAM file, you can see that it's actually a pretty complex region, right? So the reads don't map really well here. There may be a deletion here, there may not be, but part of this is probably because of the mapping. And so this is one of those regions that's not very good, or it's intractable to short read sequencing. And so these are the types of areas that can be discordant between genomes and your arrays and are hard to evaluate. The other region that's hard to evaluate are common variants, very difficult to compare. So again, if we orient, here's the Cytoscan HD probes, and this is in the DGV. This happens to be a copy number gain that's in the chromosomal microarray, and it's not in the genomes. There's no call in the genomes in the exact same sample. The log R ratio looks like it's a real variant. It looks like you can see it's all above the line. So if we zoom in on this particular area in the genome, you can see quite clearly from the histogram that there doesn't actually tend to be or any notion that there is actually a copy number gain here at all. And so what's actually happening here? If you look at this previous case, and what I've done is I've taken 3 different cases. The previous case, you can see no variant. Here's a couple of more cases. This is an additional case. It's a heterozygous deletion. You can quite clearly see that. And then here's a homozygous deletion. This deletion is actually present in about 40% of the genomes that we sequence in the population. So it has quite a high allele frequency. And so individuals who are copy number 2, like the case that I just showed you, appear as a duplication in CMA, but this is due to the baseline. So this is one of the advantages to the genome redepth methods, is that they can self-normalize across themselves and don't rely on these on baselines. So in these common variant regions, it's very hard to compare between the 2 different technologies as well. Another example is genome false positive calls that are often false negatives in CMA. So when you start getting down to low resolution, so we're trying to validate things over 10 kb in size, roughly the minimal resolution of this particular array platform. It doesn't look like there's a call here, although there may be. It certainly wasn't picked up. And if you zoom in on this region, you can quite clearly see from the histogram of the coverage, you can quite clearly see that there is a deletion here. So many CNVs near the resolution appear to be false. They appear to be false positives in the genomes, and actually they're false negatives in CMA. So this is obviously one of the advantages of genome sequencing, is that you can detect a lot smaller variants than you can in CMA, but it does make the comparison between the 2 quite difficult. This is my last example. So we're talking about clinically relevant regions. So what about large regions? And so here's the cytoband. Many of you will recognize this as the common 22q112 deletion syndrome 2.5 megabase deletion. And you can see the BAM file here. You can clearly see where the segmental duplications are here that are splitting up the call. And on the microarray, you can clearly see the call here as well, which often is sometimes overextended because there's no probes in the segmental duplications. And this is obviously the minimal region, the minimal curated region. And so if you look at this, what tends to happen, especially when you have segmental duplications, is you can't get fragmentation of your genome call. And because you're getting fragmentation, this actually makes annotation really difficult. So you can imagine that you're actually getting 3 different calls here in this region, and then you have to stitch them back together, or at least recognize that there could be 3 different calls here to be able to report it out properly from a genome. So this is one of those issues. I think algorithms are getting better at stitching these together, but we have noticed this, and it's something to watch out for. So in the final part of my talk, I just want to tell you a little bit of what we've done. We did construct a CNB Comparison True Set. We selected a lot of cases, positive controls from our chromosomal microarray analysis platform, which was the Affymetric Cyto HD. We selected a lot of the more common ones, and you can see we selected over a size range, both gains and losses, all the way from 10 kb up to variants that would be greater than a megabase in size. CNVs from the filtered, then we did, and we filtered them from the true set. So we filtered based on size, making sure that there was enough Affy probes for us to find them, greater than 10 kb in size. So this could be laboratory dependent upon what you like to report out on. We also made sure that they were as rare variants, And then we made sure that we weren't looking at variants that were had a really large overlap with some of these more complex regions. So this was our final true set. And when we did this, we did an accuracy table using the FDA comparator method, or the method that's recommended by the FDA. So the true set was rare and reportable gains and losses greater than 10 kb in size. So we had 105 of these. So using the comparator method, all the variants detected were detected by genome sequencing, giving a positive % agreement or a sensitivity of 100%. The other issue with this, of course, is specificity. Looking at on a genome-wide scale, you want to make sure you don't get too many variants. But once you filter down for rare variants that are of a certain size, you actually don't get too many. So generally speaking, at least in our experience, you're getting around 20 rare CNVs that would need to be interrogated. And then from these, only about 1 or 2 are apparent false positives per case. So we are noticing a relatively low false positive rate, and it's making it manageable to be able to report out from genomes. Just in summary, when will genomes replace chromosomal microarray analysis? Back to the question at hand, we know that genome sequencing can detect most forms of variation, has potential to be a first or single first-tier diagnostic test for individuals with suspected germline disorders. We know that some groups have already implemented low-class genomes to replace CMA, but of course, in this case, you can't really report out on other forms of variation. So here we're talking a little bit more about standard genomes, 30 to 40x. I can tell you that, at least from an analytical perspective, standard genomes can replace chromosomal microarray analysis at this time. Certainly equivalent sensitivity for rare reportable variants, and is capable of the detection of pathogenic changes that are much below the resolution of CMA. So at least in our hands, what we're going to do is for variants that are below 10 kb in size, we might have to report out on using our orthogonal method, or validate using orthogonal method before reporting out. But we haven't even broached this yet, but it is one of the advantages. I think some of the challenges with actually genome testing replacing CMAs completely is the fragmentation of calls. We saw that. We have noticed algorithm improvements over time to be able to stitch some of these calls back together. As I said, the annotation can be an issue, and when you filter out, you can sometimes lose sort of pieces of these fragmented calls. Just in terms of annotation, filtering, and interpretation, we certainly need a lot of population controls. Internal data using the exact same methods that you've been able to call with your CNVs is the most effective way to be able to filter out variants. You're essentially creating a noise database that will get rid of common and or artifacts. It's obviously technically challenging to do genomes. Informatic expertise is certainly necessary. So I think this is one of the challenges for any diagnostic lab. And it can be expensive, so a large capital cost, and even per case. This cost is coming down over time. And once you start factoring in doing a microarray and an exome on the same individual, it starts to get a little bit more equivalent in terms of cost. So in summary, I think genomes can replace CMA, and they certainly have, or there's good data to show that they have for low pass at equivalent cost and sensitivity. However, at least for standard genomes, the cost and scalability do still make it challenging for clinical diagnostic labs. And I think cost right now is probably one of the bigger challenges. So with that, I'd just like to acknowledge the Genome Diagnostics, the Center for Applied Genomics, and the Center for Genetic Medicine groups at the Hospital for Sick Children, and then the Medical Genome Initiative, which we mentioned briefly in the talk as well. Thank you.
I think we're early, guys. What are you saying? How's everybody?
So thank you, Eli. Thank you for giving me the opportunity to present our work on the detection of clinically relevant repeat expansion from whole genome sequencing. So my name is Arianna Tucci. I'm a clinical geneticist and a researcher working at Queen Mary University of London and the UCL Institute of Neurology. I have nothing to disclose for financial disclosure. So what are short tandem repeats? They are short DNA sequences that are repeated a number of times in a row. This is an example of a short tandem repeat of an STR. As you can see here, there is a 3 nucleotide sequence, CAG, which is repeated a few times over this DNA stretch. A short tandem repeat expansion occurs when the number of repeats within an STR increases beyond a certain pathological threshold, causing what is known as a repeat expansion disorder. This is the growing list of repeat expansion disorders. They are one of the most common cause of neurogenetic disorder, and they all affect the nervous system. So clinically, they present as late-onset neurodegenerative diseases, such as Huntington disease, the spinocerebellar ataxia, but also they present as neuromuscular disease, such as myotonic dystrophy or amyotrophic lateral sclerosis. They are also one of the most common cause of non-syndromic intellectual disability, such as Fragile X syndrome. Looking at inheritance, repeat expansion disorders are mostly inherited as autosomal dominant, but also as autosomal recessive, such as in the Friedreich ataxia or in canvas repeat, and also as X-linked dominant, such as the Fragile X syndrome. Looking at the extent of the expansion, so how much does an STR expand to become pathogenic, varies depending on the STR location within the genomic region. So for STR that are exonic, the expansion involves a few repeats units, whilst for expansions that are in the intronic or UTR regions, the expansion involves a few hundred, sometimes thousands of repeat units, causing a massive change in the size of the STR from normal to abnormal. The STR expansion mutation is a gradual process. Due to replication slippage events, STRs that are normal may slightly increase their size and become a bit bigger, but not enough to cause diseases. And these are alleles known as premutation alleles. Premutation STRs tend to become more molecularly unstable, meaning that in successive generations they tend to become bigger, sometimes smaller, but mostly bigger, and expanding into the full mutation pathogenic range. So the gold standard to detect short tandem repeat expansions are PCR-based methods, namely fragment analysis and RT-PCR. For larger expansions, Southern's is also employed. In the last few years, a few methods have been developed to call short tandem repeats from next generation sequencing. And the work that I will present here is based on a tool called Expansion Hunter. But in principle, they all work in the same way. So how does the detection of STRs from whole genome sequencing works? So the software will scan a BAM file, so for example, a whole genome sequencing files, to detect reads that contain the repeat fully. So these are the spanning reads here on the left, or partially flanking reads and in-repeat reads. Meaning that from reads that fully contain the repeat, and an exact repeat size can be estimated. But while from reads that only partially contain the repeat, a confidence interval for the repeat size is estimated. So this means that for STRs that are shorter than the read length, an exact repeat size can be estimated, as opposed to STRs that are larger than the read length, where the size can only be estimated with a confidence interval. So this slide shows you the relationship between STRs in different loci, the repeat length, and the sequencing read length. So on the y-axis, we've got the different short tandem repeat expansion loci, so Huntington disease, Friedreich ataxia, SBMA, and so on. On the x-axis, the repeat length. Alleles are colored in different colors. So in blue, normal alleles, yellow, premutation alleles, and red, full expansions. And the black dotted line represents the whole genome sequencing read length that most widely adopted, which is 150 base pairs. And what this slide shows you is that for the majority of the repeat expansion disorder, loci have strs in the normal and permutation range smaller than the read length, meaning that they can be accurately sized. So going on to our study, the aims of our studies were two really. The first one was to test the performance of a pipeline to genotype short tandem repeats from whole genome sequencing. And the second was to apply the pipeline to patients that had molecularly undiagnosed disease. We carried out this project within the 100,000 Genomes Project. As most of you will probably know, the 100,000 Genomes Project is a UK government-led research initiative to test the benefit of whole genome sequencing in healthcare. And for that project, about 85,000 patients from the National Health Service were recruited if they were affected by a rare disease or cancer. So those patients had their whole genome sequencing, but also the data set comprises clinical data which were collected alongside the patient samples at the time of recruitment. In the project, patients that were affected by rare diseases were recruited alongside their affected and unaffected family members. So for the performance of the pipeline, obviously, aim one was to compare the gold standard, so PCR-based code, to whole genome sequencing codes. And in order to do that, we built a data set of negative and positive controls. So for negative controls, we've collected PCR test data on 100,000 Genomes Project participants, patients, that were tested prior to recruitment to the project and therefore mostly carried alleles in the normal range. We've also collected
This to do with our internet. Why, George, is your internet down? Hey guys, can you hear me still? Is it my internet that's down or is it hers? Oh, it's up again. That's the volume.
Majority of alleles that were predicted to be normal BGS, and similarly for PCR predicted alleles predicted to be expand.
Hey George, is your internet going down? What is going on with this?
97% and a specificity of over 99%. We went on to investigate false positives and false negatives scores by visually inspecting the reads that were used by the software to estimate repeat size across all performance data set. So this is an example of an STR in the androgen receptor locus. The genotype predicted by WGS was 28 and 28, so by PCR is 28 and 28 again. So this, as you can see here, this is a grouping of all the reads that contain the repeat, and if you could count the number of CAG repeat, it will go back down to 28.
Oh my gosh.
That's between the genotype predicted by WGS and genotype predicted by PCR. Looking at a sample that was discordant, again, a call in the androgen receptors locus, genotype predicted by WGS 28 and 52, genotype predicted by PCR 28 and 28. At the closer inspection of the reads that were used to support a genotype predicted of 28. We see here again a large number of reads supporting 28. Going down to the bottom of the graph, we can see the visual representation of this long reads, which is the one that has been used to estimate a genotype of 52 repeats in this sample. If you look here, you can see that most of the nucleotides here are in lowercase, and these are nucleotides that have bad sequencing quality. So we can discard these reads and actually correct after visual inspection this call by WGS and say that actually the evidence from this WGS call is that the genotype is 28 and 28, like PCR said. So we performed a blinded visual inspection of all calls included in our performance dataset and showed that after visual QC, we were able to correct all of the false positives and the majority of the false negative scores, further improving the sensitivity and the specificity of the pipeline. Here, I would like to say that 2 out of 3 of our false negatives are from genomes that were sequenced at 125 base per length, as opposed to all the others that were sequenced at 150. So that may be having an effect there. Looking at the breakdown of the performance by locus, we've got here on the y-axis all different repeat expansions, loci, androgen receptor, 80 and 1, ataxin 1, 2, 3, 7, and so on. And on the x-axis, the repeat size. The background color represents the color of each alleles predicted by PCR, and the colors of the dot represents the estimate performed on that alleles by WGS. So a blue dot over a blue background represents a true negative scores. A red dot over a red background represents a true positive scores, while mismatches such as this one, a red dot over a blue background represents a false negative here, and the other way around, this is a false positive. So this slide shows you again that the ability of the pipeline to distinguish between normal and abnormal alleles is good across all of the repeat expansions loci. But also what this shows you here, for example, is that all repeats that were predicted by PCR to be in the full expansions range for FMR1 were predicted to be in the pre-mutation range by whole genome sequencing. So whole genome sequencing constantly underestimates larger repeats. Looking again at the repeat size prediction, comparing the repeat size by PCR here on the y-axis and the repeat size by WGS, and dividing the alleles into alleles that are shorter than the read length, so 150 base pairs, and alleles that are larger than the read length, we can see that the correlation is quite good between PCR and WGS for alleles shorter than the read length. But what we see here, what I've shown you before, is that the majority of alleles larger than the read lengths, for example, the one in FXN or FMR1, tend to be underestimated by WGS. So to conclude, for this first part of our study, we can say that the pipeline can distinguish between normal and expanded alleles with high specificity and sensitivity. Furthermore, the visual inspection improves the performance, especially to identify alleles with poor sequencing quality or mapping quality, and therefore improves the performance of the pipeline. Size, with regards to size prediction, the WGS pipeline can size normal alleles, provided that they are smaller than the read length, but cannot size large expansions. So this will affect the sizing of expansions, for example, in FMR1, FXN, DMPK, or C9orf.
Boopity boopy. Oh gosh. Yeah, with the next presentation after this, man, I can't wait. Excited.
To the 100,000 Genomes Project. Patients, as I said before, this is a large whole genome sequencing data set together with clinical data.
This is interesting as well. If you look at this, what she's presenting for
patients that were thought to have a phenotype consistent with the repeat expansion disorders. So we analyzed all of those patients that were recruited under a late onset neurodegenerative disease, such as hereditary ataxia, spastic paraplegia, Parkinson's, dystonia, ALS, and so on, but also analyzed patients recruited under intellectual disability or a neuromuscular disease. So STR diagnosis. By STR diagnosis, I mean we've detected a number of STRs expansions in patients recruited with a phenotype potentially consistent. We validated it by PCR, and then we've contacted the clinician that recruited the patient to the project to tell them there is an expansion here which has been validated. This could be a diagnosis. And so the clinicians would order the standard NHS test to issue a diagnostic report. So what I like to show here is that for diseases that are genetically heterogeneous, such as hereditary ataxia, the pipeline was able to detect pathogenic diagnostic repeat expansions across a range of different STR loci, such as ATN1, Ataxin2, 3, 7, CACNA1A, and so on, showing that this pipeline may be particularly suitable for genetically heterogeneous diseases. Equally, we detected expansions in loci that presented with a wide phenotypic range. So these are expansions in the androgen receptor, which causes Kennedy disease, which is a form of motor neuron disease. We've detected the expansions in patients recruited under ataxia, but also under Charcot-Marie tooth, which is neuropathy, and some under amyotrophic lateral sclerosis. This again shows you that the pipeline can be also useful for loci that present with phenotypically heterogeneous features. Strikingly, we've detected rare events of STRs that typically cause diseases in the adults, in children. This is a known phenomenon known as anticipation. We know that for some of the short tandem repeat expansions, the larger the repeats and the earlier the age of onset and the severity. So this plot shows you here the allele count versus the repeat size of 4 short tandem repeat expansions that typically cause diseases in adults: ATN1, Ataxin2, 7, Ataxin 7, and Huntington. The vast majority of allele we detected were in the normal range, but as you can see here, we've got here the white triangle, some pathogenic expansions detected in adults, and some very large pathogenic expansions detected in the pediatric population. And what's really striking about this result is that most of these families where we had the pediatric presentation had a negative family history of the disease at the time of recruitment. So this is an example of one of the child that carried a very large expansion in Huntington. The repeat has been shown to jump from one parent that carried a repeat, which was smaller than the pathogenic range. This was, I think, 38 repeats. Again, showing you the phenomenon that I've talked about of molecular instability of larger STRs. We've also detected some unexpected diagnoses, such as expansions in SCA36, have been previously described in Asians, so Japanese population, and Hispanic populations, but were thought to be absent in the UK. However, we've detected a number of families with expansions in SCA36. They are all from the UK. This plot shows you the pile-up graph by expansion hunter, showing you that there are a large number of reads with the repeats supporting the presence of an expansion. And this is the RP-PCR that shows you the typical shape that the RP-PCR has when there is an expansion there. What we can't tell here is how large the expansion is, and we are trying to confirm this by Southern as well. So to conclude, I've shown you that we've tested a pipeline to call short tandem repeats. This pipeline has high sensitivity and specificity, and can also size alleles in the normal range, provided that they're smaller than the read length, whilst it cannot size alleles that are in that are expanded. And this, the size prediction particularly affect very large expansions such as FMR1, C9 North, and the MPK. The pipeline can detect pathogenic expansion in patients and is particularly suited for genetic and phenotypically heterogeneous diseases. We've tested, obviously, 13 of over 40 short tandem repeat expansion loci, so more work will be needed to test this pipeline on other loci. With this, I would like to thank all of the people that contributed to this work, and specifically Christina, who works at Genomics England, and Queen Mary with you, who has carried out most of the analysis, and also a thank to the patients and families who were recruited to the 100,000 Genomes project. So thank you.
Very interesting. There's some parts in that I found very interesting. I'm going to piece it together for you. She just said thank you for Queen Mary's.
Hello, I'm excited to be with you today and to share some of our recent data and some data from a recent publication. So the title of my talk is Clinically Relevant Genes Hiding in Plain Sight and How Long-Range Technologies Resolve Them. I have nothing to disclose. So for years, we've known that there are certain regions of the genome, or what we might call dark. But in our research, as we started exploring certain genes, we started to bump into some really important genes that were dark or that are overlooked, and realized that this is a bigger problem than anyone had given credit for. But there are two basic types of darkness that I want to introduce. The first is what we call dark by depth, and the second is dark by mapping quality. So the first one, I'm going to show an example of the gene HLA-DRB5. And if you've never looked at a what we call a pile-up of sequencing reads, this is what it looks like. So let me orient you. All of these reads have been aligned to the gene HLA-DRB5, and each little gray bar that you see here represents one of those reads. And what you typically want to see is a nice, deep, rich stack of gray bars that represents how deep the coverage is. And in this sample, it's approximately 60x coverage. But as you get deeper into the gene, you start to see fewer reads aligning. And in some regions, you see 0 reads. So this is an example of dark by depth. There are regions where there just are no reads to be aligned. The second type of darkness is what we call dark by mapping quality. And in this case, we're looking at HSP-A1A. And as you enter this gene from the left, you again have a nice deep rich stack of well-aligned reads. And then you see these transparent reads, which are reads that quote unquote did not align well. But then as you exit the gene, you again see a nice deep rich stack of aligned reads. So in this case, what's happening is, for some reason, the bioinformatics software that we call the aligner that aligns these reads to the reference genome feels or determined that these reads did not align well. So what's happening here? Well, I guess first, let me tell you the end result of both of these situations. In either case, whether it's dark by depth or dark by mapping quality, when you get your final list of variants, or your VCF file, if you're familiar with those, any variants within these dark regions are completely missed. And of course, you're not made aware of this. So you have no idea that these regions are being completely missed. So that's a big problem, especially when we're dealing with genes of clinical significance. So let me explain how dark by mapping quality happens. It turns out that dark by depth is a rare issue. It's mostly the dark by mapping quality. And it's actually what we term camouflaged regions in our paper. So what's happening in this case is HSP-A1A and HSP-A1B are two genes that are just down the road from each other on the genome. They're about 14 kb apart. And that means that there's also a large portion of them that are identical. It's approximately 50% of the genes are identical. So when the aligner gets one of these short reads, these transparent reads, that aligns equally well to either gene, it doesn't know where to put it. So it randomly assigns the read to one of the locations and gives it a mapping quality of 0. But as I mentioned, the end result is that all variants in either in this region or this region for both genes are completely ignored. They're lost. So as I mentioned in our work, we started to bump into some really important genes for our research that were dark or camouflaged. So we decided to systematically go across the genome looking to determine how many genes are affected. And it turns out that over 6,000 gene bodies are at least partially dark. And over 500 of those are completely dark from end to end. So here on the left, I'm showing you a basic karyotype. You can see that where each little hash represents a gene that's partially dark. And you can see they're scattered throughout the genome. On the right, I'm showing a plot where the y-axis is percent darkness for a given gene. And the x-axis is rank order for those genes. So the first set represent the 527 that are completely dark. If you do a 5% cutoff, there are at least there are over 2,100 genes that are at least 5% dark gene bodies. But you may have noticed that I'm saying gene bodies because there are different types of gene bodies. And so we wanted to know which types of gene bodies are most represented here. And we were surprised to find that most of them were actually protein coding genes, followed by pseudogenes and various RNAs. I would have expected, quite the opposite, that fewer protein coding genes. But from there, we then wanted to look and see which element of a gene body is most likely to be dark or most represents the darkness. And unsurprisingly, it turns out that it's the introns, followed by exons from non-coding RNAs, then actual coding exons, followed by 5' and 3' UTRs. Now, I hope this is a group that I don't have to persuade of the importance of introns. Introns are biologically critical, and a large proportion of disease-causing variants are actually intronic. So a lot of researchers will quickly discount introns and say, who cares if the introns are dark? But that's not true. There are a lot of intronic variants that cause disease. But just to demonstrate that we're not trying to be alarmists, I'm going to focus only on the protein coding regions for the next little bit. So if we look at just the coding regions, the CDS regions, there are almost 600 genes that are at least 5% dark in coding regions, and 117 of them are completely dark, the entire coding sequence. So from there, and this is a slide that I'm really excited to share with you all, especially with this group. We wanted to know, okay, what's the clinical significance of this? How many of these genes that are at least 5% dark are already known to be involved or associated with disease? And we found 76 genes that are at least 5% dark that have mutations in the Human Gene Mutation Database, which is a manually curated database, and they're associated with 326 unique human diseases. And some really big hitters here. So if we look at RPGR, it's associated with various ophthalmic diseases, TBX1, various cardiovascular defects, coronary artery disease, hearing loss, various neuromuscular disorders, various developmental or neurodegenerative conditions, spinal muscular atrophy, and even Alzheimer's disease. And those last two were the ones that were really eye-opening to us because of our research involving ALS, Alzheimer's disease, and frontotemporal dementia. So, and that's not to even, what about those 117 genes that are completely dark? Unsurprisingly, we know nothing about those. We have no variants. You won't find them in ClinVar or other databases because we don't get them. What significance do those have? That's a big open question. So I want to show you a couple of, I guess, dig in a bit and show you a couple of examples. Specifically SMN1 and SMN2 here. They are over 90% camouflaged, and they're kind of like the HSP A1A and B example that I showed you before, where they're camouflaging each other because of a gene duplication. But what I really want to show you here is that this isn't just an issue in our data, right? I didn't just handpick some of our some bad data, or it's not that we had some sort of technical issue. The plot I'm showing here is a coverage histogram from the NOMAD consortium. If you're not familiar with that, it's an aggregate of almost 150,000 exomes and genomes. And you can see that the coverage for the majority, almost 95% of SMN1 is 0. And green line represents the genomes, the blue represents the coverage for exomes. So this is a big problem. And SMN, as I mentioned, causes spinal muscular atrophy and has even been implicated in ALS, which is why it's of interest to us. Looking at CR1, I told you that's a favorite gene for me with my interest in Alzheimer's disease. I've been involved in Alzheimer's disease genetics for 8 or 9 years now. And until we bumped into this, I had no idea that 26% of CR1 is camouflaged. And interestingly enough, it turns out it's the binding domain, the C3b, C4b binding domain, which is part of the complement cascade process, which is a primary hypothesis in Alzheimer's disease. So really shocking here. And then, all the other genes that we showed. I also wanted to briefly show ARX. It's about 13% dark in whole genome data sets. If you look at the green line, you're going across the gene. It's complete coverage. But then in this one region, it dips. But I'm showing here not a histogram of the coverage. I'm showing the percentage of samples in the NOMAD consortium that have sufficient coverage. And it turns out, for some reason, 70% of them have good coverage or meet our minimum requirements, but 30% of them are still dark. And we think that might be DNA quality or library prep, but we don't have access to that data to determine. But I also want to point out, if you look at the blue data, that's the exome data. The camouflage issue is dramatically magnified in exomes versus whole genomes. And that's really important to know for, in clinical genetics. So hopefully to this point, I've convinced you that there are a lot of really important genes that are dark, most of them camouflaged, at least portions of them. What we would consider a significant portion, at least 5% of the coding regions, plus a bunch more with intronics regions that are dark. So we're intentionally trying to understate the problem. So the problem is actually bigger than what I've been showing. But what are we going to do about it? In our paper, I'm not going to go into this, but we developed a method to rescue mutations in short read data. But I want to make it clear that we do not consider that a solution. That is simply a Band-Aid. What we need to do is be looking for ways to resolve this going forward the right way, and that's with long-range technologies. And let me show you how these resolve these dark regions. So again, looking at just the regions that are at least 5% dark encoding regions, so those genes. If we take Illumina, or excuse me, no, this is genome-wide. This is all of the dark regions. We take the Illumina reads as our baseline, and then we compare other long-read or long-range technologies. So 10x Genomics, their synthetic long reads are no longer available, but they were at the time. And then there's PacBio and Oxford Nanopore Technologies. So as you look down this this table, the lower the number, the better. So basically, what this is saying is that 10x Genomics synthetic long reads resolved about 49% of all of the dark regions. PacBio resolved about 64% of all of the dark regions genome-wide. And Oxford Nanopore Technologies resolved about 91% of all of the dark regions. And this is a plot of the rank order in percent darkness that we showed for each technology. So again, showing Oxford Nanopore Technologies resolves most of them, leaving very few regions unresolved. So I want to show some specific examples of this, so you can kind of relate and see what we're talking about. So if we go back to SMN1 and SMN2, we look at Illumina, you can see these big gaps for SMN1 and SMN2. 10x Genomics in this case resolves it perfectly. PacBio technically resolves it according to our criteria, but if you look at this histogram of coverage, you see that it drops dramatically in the middle of the genes. Oxford Nanopore Technologies goes through them without issue. If we look at CR1, again, Illumina, you see the big gaps. In this case, 10x Genomics has no effect. With PacBio, again, it technically resolves it, but you see this dip in coverage. With Oxford Nanopore Technologies, you do see a minor dip, but again, the coverage stays very, very high throughout. So to this point, I've shown the long range technologies that, the long range sequencing technologies, but there's another technology that I want to introduce that has a lot of value, and that's the Bionano Genomics saphyr. It's not technically sequencing; it's optical DNA mapping, and it's all of these technologies are excellent at identifying structural variants, and structural variants are critical and are a major player in disease, as I will demonstrate here. So, for example, ALS is caused by a C9orf72 G4C2 repeat expansion. And I'm showing here a southern blot of DNA of this repeat expansion, where we have a specific case that has the normal allele and then two different bands of expansions, a smaller and a larger one that is beyond our ability to size using southern accurately. And it's even would be difficult to size with other long read technologies. But using the saphyr, I'll just briefly explain here on this green bar represents the reference genome, chromosome 9. This is where the C9orf72 gene is. Here, basically what you're seeing are 5 different haplotypes that we were able to resolve, showing the mosaicism of this repeat expansion within a single piece of tissue, brain tissue. In this case, it was the frontal cortex. And those ranges, those sizes ranged from 2 kb, this band, all the way up to 31 kb, this band. Really just a very powerful way. We can now directly size this repeat expansion. And we even had individual molecules where we were able to size up to 45 kb, which is just remarkable. Let me show you another example. This is from an alpha-synuclein triplication that is known to cause Parkinson's disease. Of course, both of these examples are structural variants. So if you're into copy number variant analysis, with the saphyr, you immediately get this data where as we're going along the chromosome 4, you have 2 copies, and then you see this sudden spike to 4 copies, and then back down to 2 copies. So we immediately see the breakpoints. We see that it's a 2 megabase region that has been that now has 4 copies. If we look at the raw data, we're actually able to resolve the entire first normal germline haplotype. That's what this blue bar represents. You can see individual genes represented here, and this is where alpha-synuclein is. On the second haplotype, we're able to resolve that basically there's an inversion and a triplication. So what you wind up with is alpha-synuclein, 3 copies of alpha-synuclein on the second haplotype. So up to this point, no one had resolved the genomic structure of this. But we were able to do that with these long-range technologies. And I want to emphasize that we're not just a single haplotype, but both germline haplotypes, which is really powerful. So I know that there have been a lot of criticisms with, or concerns maybe, with long-range technologies. And so the question might be, when is this ready for research or clinical use? Well, I'd say for research, it was ready, yesterday. It's been ready for a long time. For clinical, I would say it's ready now, in my opinion. And so I want to address some common concerns. So, sequencing error rate is probably the most common concern that I hear. I would say that's no longer an issue. With PacBio, they have circular consensus sequences, and you can use overall consensus that you easily get well above 99% individual nucleotide accuracy. Similar with Oxford Nanopore technologies. The cost per sample has been dropping dramatically over the years. For whole genomes, it might still not be feasible, but you can do targeted approaches, such as using Cas9 or directed primers. And then with Oxford Nanopore, you have similar options. Plus, they have several instrument options for various needs, including an upcoming Plongle, which is directed specifically for clinical users. For the Saphyr, it's really easy to get sample pricing down to $450 per sample for 400x coverage minimum. I mean, that's really the low bar. It's actually quite easy to get well over 1,000x coverage genome-wide with the Saphyr. For the bioinformatics, these days the tools are mostly available, and for the Saphyr, it's straight up turnkey. And in fact, they have some. There are clinical tests available now using that technology. Mosaicism is a really strength for these using either targeted or consensus sequencing is now possible. So really, long-range technologies are going to improve diagnostics, and they're going to enable us to have a greater understanding of disease etiology. And I just want to emphasize that that not only are we able to get small mutations with these these technologies, we can also get structural variants, which are critical. And I want to share a list of diseases that are caused or either directly caused by structural variants or structural variants are known to be involved in them. Parkinson's disease, I showed you a specific example. FSHD, there's a repeat retraction actually. There's a clinical test available for that. Fuchs disease, ALS, FTD, all repeat expansions. The Fragile X, repeat expansion, leukemia. There are 12 distinct spinocerebellar ataxias with 12 distinct expansions that cause them. Autism, Crohn's disease, schizophrenia, Huntington's, of course, that's the classic example. So critical benefits for long-range technologies, you can resolve not just point mutations, but structural variants, and these are really important. You can resolve entire haplotypes, both germline and mosaic haplotypes. So we're not just a single haplotype where we're trying to discern them, which gives us a lot more power to understand disease. We can actually resolve the entire major histocompatibility complex, a 4 megabase region that is very difficult, but we can do that. Also using these technologies, Drs. Philippi and Miga et al., they have done the first telomere to telomere sequencing of the human genome. They published the X chromosome. They have more coming. It's really remarkable. And they used all of these technologies that I've been talking about today. Thank you. I really appreciate your attention today, and I'd be happy to take any questions.
Oh, baby. Oh, baby.
All right. Thanks, everyone, for
Q&A
your
now.
attention during those pre-recorded sessions. So what we'll need to do now is, as you can see, we have our speakers back in the live format. And I think what we'll go to now is we'll just jump into some case examples. There's a number of questions in regards to size and those kinds of things. Let me step back for a second. And just say that I know there was some resolution issues during some of those talks. So we apologize for that, but just know that all the talks are recorded and the quality of the recordings are very good. So you will have opportunities to watch them again at a later time. I will also say that there's been over 400 participants for this entire session, so that is fantastic. And I think that's probably contributing to some of the streaming problems that we maybe have been having. And it's also led to an abundance of questions, and I will do my best to try to address at least the major themes of the questions as we go through this live portion. But if I don't get to your question, I apologize, but please feel free to reach out to the speakers or try to find them during the meeting for the remainder of the meeting. So what we'll do now, I think, is go into some case examples and really see how some of these applications of genome sequencing and long-read sequencing technology can perhaps fit into a clinical workflow. So I'll turn it over to our speaker, Dr. Tucci, if you want to kick things off with the case examples.
Yeah, thank you very much. So we've got a couple of cases really that illustrate the ability of genome sequencing to resolve complex clinical cases, but also how the limitations of whole genome sequencing can be addressed by newer technologies such as the optical genome mapping and long-read sequencing. So on to the first case. This is a 73-year-old white British male presented about 10 years earlier with a history of progressive balance problems. In the past medical history, he suffered from thyrotoxicosis with apparent myopathy, age 60, and he also had high tone deafness since age 40. In his family history, he had one older sister who had problems similar to him, so balance and hearing problems, two sisters who were well, two sons in their 40s were also. On examination, he had bilateral ptosis, ophthalmoplegia, and poor upgaze. Hard cerebral signs were noted, including cerebral ataxia, finger-nose ataxia, and ataxic gait. The motor examination revealed mild proximal weakness. He had wrist reflexes, and the sensory examination was normal. The brain MRI revealed mild cerebral atrophy. So at this stage, mitochondrial disorder was suspected. So he had a muscle biopsy that showed some cytochrome oxidase negative fibers and long-range PCR showing multiple mitochondrial rearrangements. So he was testing using the standard of care genetic testing available in the NHS. So he had the mitochondrial genome DNA sequencing, which was normal, mitochondrial nuclear gene panel, which was normal, and it was also tested for the most common cause of adult ataxia. So the spinocerebellar ataxia 1, 2, 3, 6, 7, and 17. These are all repeat expansions, and they were all normal. So he was recruited to the 100,000 Genomes project. We analyzed his genomes. We applied our short tandem repeat expansion pipeline, and we detected more than 36 GG-CCTG repeats in NOP56. The plot here shows you the pile-up, so the whole genome sequencing from this patient in the NOP56 region, showing a short allele and a larger allele, indicating the presence of an expansion in NOP56. Expansions in NOP56 cause spinocerebellar ataxia 36, and on review of the clinical cases, its features were consistent with spinocerebellar ataxia. The expansion was also confirmed by RP-PCR. So what do we know about SCA36? We know that it's a rare form of autosomal dominant hereditary ataxia characterized by cerebellar ataxia, sensory neural hearing loss, and motor neuron impairment. As we said, expansions in a hexanucleotide, GG-CCTG, in the intron 1 of NOP56 cause SCA36, with the normal alleles be shorter than 14 repeats and larger alleles being larger than 650 all the way up to over 2,500 repeats. SCA36 has been described in about 100 individuals, mainly from two regions, Western Japan and other parts of Asia and Spain. But most importantly, it was thought to be absent in the UK because a previous screening about 5 years ago of patients with ataxia in the UK was negative. So there isn't a diagnostic test for SCA36 currently in the NHS. So we went on to analyze the SCA36 expansion in patients recruited to the 100,000 Genomes project, and this is really a nice example that shows you how whole genome sequencing can quickly lead to a diagnosis. So we analyzed over 1,182 families recruited under hereditary ataxia, analyzed whole genome sequencing data using our short-time repeats pipeline employing the expansion hunter, and when we detected an expansion, we went on to validate it by RT-PCR. So we detected a total of 6 families with not 56 expansions and a phenotype consistent with SCA36. And of note, all of these families are from a white British background, confirming that the SCA36 does exist in the UK population. Going on to the second case. This is a 70-year-old female that was seen by neurology because of decline in short-term memory since age early 60. She suffered from episodic memory problems. She had limited vocabulary, but occasionally word-finding difficulties. Behaviorally, she was noted to have become a bit more irritable, but the behavioral problems had not been progressed for about 10 years. Past medical history was unremarkable. In the family history, her mother was similarly affected with memory problems, started in her mid-60s and a history of wandering. The MMSE examination, she scored 26 out of 30, and the general neurology examination was normal. The MRI scan showed significant central atrophy, particularly in the temporoparietal area. So at this stage, a query diagnosis of probable young onset Alzheimer's disease with some atypical feature was suspected. Again, here, standard of care genetic testing available in the NHS was performed, and this was the dementia gene panel comprising about 18 genes that are involved in dementia, and this was normal. She was recruited to the 100,000 Genomes project. Again, we analyzed her genome, and an expansion in C9orf was detected. Again, the pile-up plot shown here in this part of the graph, showing a short allele and a larger allele in C9orf72. Expansions in C9orf72 cause frontotemporal dementia, and again, upon revision, her phenotype was thought to be consistent with that, and so a genetic report was issued. So in both cases, I've showed you how whole genome sequencing allowed us to reach a diagnosis first in a group of families with SCA36 and here in this patient. But what is the limitation? The limitation really, as I show you probably, and you've noticed in my other presentation, is the size. So this is just a bigger example of the pile-up graph showing the expansion in the patient that carry the expansion in C9orf. Here you see the hexanucleotide repeat a few times, 5 times. These are the reads that support the short allele, so genotype of 5. In the lower part of the graphs, we see a large number of reads with many hexanucleotides that go up to 25, 25 being the limitation, which is the read length. So we are limited in the size, in our ability to accurately size repeat expansions by the sequencing read length. And this is a problem that can be addressed by other technologies, and now Mark is going to talk about those, so I'll pass it on to him.
Great, thank you, Dr. Tucci. Those were great examples. So I hope that, among other things, that we've been able to show how important structural variants are in clinical disease. Earlier in my talk, I showed some data based on C9orf72. This is that slide showing you again the southern blot with the C9orf72 expansion carrier. And I just wanted to show some additional data highlighting how long-range technologies can actually help us size repeat expansions and other insertions. They actually turn out to be some of the most challenging to accurately assess, because you have to have a read or an optical map that can span the entire insertion or repeat expansion in this case. Next slide, please. So I showed you this before also. So basically, we're showing the mosaicism and the continuum of repeat expansion sizes within a single piece of brain tissue. Next slide. But digging in a little bit more, this is that same sample. So looking at the top, you see the blue horizontal bar that represents the normal or the normal allele, the reference allele, according for this individual haplotype 1. And the green horizontal bar represents chromosome 9. You can see that the first haplotype aligns perfectly with the green bar of chromosome 9, based on the perpendicular gray lines going between them. And then below the green bar, you can see a second haplotype that in this case is just an average of all the haplotypes. There are various ways you can perform this analysis, and in this case, we're just sort of forcing the data into 2 haplotypes. So below that second blue bar, you'll see individual optical maps for individual DNA molecules, and that's part of what's really fascinating or remarkable about this technology is that we're not just looking at a glob of DNA. We're actually able to measure individual DNA molecules. And what I'm trying to show here with the black arrow is the continuum of repeat expansion sizes. So as you enter from the left, each little blue hash indicates a motif that you should observe, and they align perfectly with the reference genome in green. But then once you reach a certain point, you start to see these larger gaps. Next slide. And the largest expansion size that we were able to measure in this individual is actually 45 kb, which is remarkable. And that just tells you just how large the DNA molecules are, how high molecular weight this DNA is, to be able to size a 45 kb expansion. And this is just kind of a proof of principle and how this applies not just to CNR72 and ALS and frontotemporal dementia, but to other diseases and the ability to size them accurately. Next slide. So I wanted to also show some data from a previous publication. What I have been showing you is not published yet. But this is from a 2018 paper where we were comparing PacBio and Oxford Nanopore technologies to see if either or both technologies could sequence through the CNR72 G4C2 repeat expansion. And the short answer is yes, they do. And what I'm showing here is a pileup of PacBio data, but Oxford Nanopore data would be quite similar. And we're able to get 4 reads that either went deep into the repeat expansion or actually spanned the expansion. One limitation with this is how large of an expansion we'd be able to size with PacBio specifically, because there are some physical limitations in their platform. And when this paper was published, if you're not familiar with it, at the time we were not able to sequence through a patient-sized repeat expansion with Oxford Nanopore, but that has since been resolved. The various things that we've learned and that have been improved, they're continuously improving the platform. So now there's no issue getting through that. Next slide, please. And wanted to lastly show an example of targeted data using the Cas9 approach with PacBio, where this is again from an actual patient sample, where we were able to observe a distribution, again confirming the mosaicism, the varying size of the CNR72 repeat expansion. And in this case, the max size that we observed was about 6,000 nucleotides, which well represented the patient sample. So I hope that this this data was helpful. I personally really enjoyed Dr. Tucci's and Dr. Marshall's talks. Very informative, and of course, we'd be happy to take any additional questions now.
Great, thanks, Mark and Ariana, for that, going through some case reports. So I guess I'll kind of start with, in thinking about, I mean, Ariana, I think you showed that repeat expansion can be detected by a whole genome sequencing. And Mark, we can size those very effectively with the long-read sequencing, but what do we need to do to incorporate that into clinical practice? Do we need to be confirming these, or what do we need to do? I guess, Ariana, I guess we can start with you.
Yeah, obviously it's very important that that the PCR confirmation is performed after the whole genome sequencing call has been visually confirmed. I mean, this is the very first shot at it. We had a large data set where we were able to confirm that whole genome sequencing can detect these expansions with high sensitivity, but I think it's going to be a very long while before we will move away from PCR. So definitely required PCR confirmation. Yeah.
And do you think we need to know the size of these repeats? Do we know the clinical relevance of these large expansions?
Yeah, exactly. The question is whether when the size when we need to know the size when there is a clinical reason to know the size. So, for example, for Huntington disease, we have to be able to accurately size repeats that are in the pre-mutation or reduced penetrance range, because clinically they mean totally different things. Whilst for repeats that are very large at present, most of the clinical diagnostic tests only detect the presence of the expansion, for example, by RT-PCR, and can only give a confidence interval for the size. So I think we will need to do more long-read sequencing to understand what the clinical implications are for us to know exactly what the sizing mean for these large expansions.
All right, and a question for you, Mark. Some of the data you showed that even with the long-read technology, we're still getting about maybe 10% of the genome, I think you said, that is still dark. How do you explain that, or what can we do to overcome that?
Yeah, so some of those regions are still just really quite large, and really would require even longer reads than were available. And the technologies are continuing to improve, but it's just based on the improvement from short read sequencing to long read sequencing. There's really so much to gain. And of course, ultimately, we want to be able to get through all of it. At this point, in order to do that, it generally takes multiple technologies or multiple runs of the same technology, sort of emphasizing you can take different approaches. So, for example, with Oxford Nanopore Technologies, you can take an approach that does ultra long reads, but with that, you get shorter, or I should say, lower depth. Or if your situation requires greater depth, then you would prepare a library of shorter reads. And so there's this continual push to, of course, be able to do everything in one sequencing run, but I don't think we're there quite yet. But for the majority of situations, there is a technology or an approach that will cover these regions.
Great. Right, going back to Christian's talk, we know that exome sequencing is pretty commonly used in the clinical setting. Why do we need to move to? And we know that can cause copy number variation with pretty good reliability. So I guess advantages, disadvantages of genome versus exome for copy number variation detection.
Yeah, thanks. It's a great question. So thanks to whoever asked. I mean, with exome sequencing, I think that whole enrichment process or the capture really introduces a whole other set of issues, right? And And it's another set of variability. And what you really want when you're calling copy number variance is pretty uniform coverage if you can get it. So genomes, because they're a lot more uniform in general in their coverage, tend to be a bit more sensitive. It's absolutely true that exome sequencing, copy number variation analysis has come a long way. It's very good at detecting larger variants that would be much more in the pathogenic realm. But I think where genomes excel is the smaller variants. We've observed things under 3 to 4 exons are not well detected in exomes, and that's where genomes tend to excel. And of course, the other thing is that you know where the breakpoints, because a lot of the breakpoints will be outside of regions that are captured by the exome. So that tends to be where it's excelling. And those single exon deletions, duplications with genomes is very accurate compared to exome sequencing.
And then kind of following up on some of the technical aspects of that, do you have an experience with mosaicism from whole genome sequencing or whole chromosome loss deletions? What's your experience with whole genome and those types of things?
Yeah, so one of the weaknesses of genomes is depth, I guess, is that, at least we know for single nucleotide variants, mosaicism sometimes is called, or will be better called, in an exome because your depth is much higher if you're going to, 100x versus 40. For CNVs themselves, the larger ones are okay. You can call them because often you can start using more like a Beal allele frequency to start looking at where you can tell that there's a mosaic change. So it's true that it's a little less sensitive for mosaics. I haven't had a lot of experience with it. We've only had a few of them here, but we've been able to see them. But it's been mainly from the data from the SNP calling data and looking at the allele fractions. Whole chromosome is okay as long as you know what you're looking for. I think again, the SNP allele fractions can be used here to be able to find these. Because the genome is self-normalizing across the chromosome, that's where you can get into some issues with aneuploidies. But in general, as long as you take that into account, it's accurate.
Great. Question for Ariana. Can you comment a little bit of quite a few questions on different expansion repeat callers that could be used? Can you comment a little bit? You used Expansion Hunter. Do you have any experience with other software or approaches?
Yeah, well, we started this work in 2017, and by the time only 2 softwares were available, which is one called Hipster and Expansion Hunter. And so because Expansion Hunter was the only one that was able to genotype repeats that are larger than the read length, and because some of the expansions in the pathogenic range are larger than the read length, then we've decided we're going to use Expansion Hunter. Now, obviously, since 2017, a large number of repeats, and we have done a little bit of benchmarking with the next version of Hipster, which is called Gangster. We saw similar results, except Gangster had was a little bit less, had less sensitivity for large expansions such as C9 morph. And I think going forward and thinking about WGS implementation, I think when thinking about a short tandem repeats pipeline, it's important to choose a tool that A, can genotype a repeat that is larger than the read length, because these are in the pathogenic range, and B, it's able to genotype and to tell the number of each allele, even though it is an estimate. This is because alleles that are shorter than the read length need to be accurately sized, because, as for example, for Huntington's, they are really important. It's really important to know the exact size.
Great. I think we're just about out of time here, so I just want to make a couple comments. There were a lot of questions, of course, about reimbursement, implementation of genome sequencing in a clinical setting, and I'll just say that there is a session tomorrow, I believe at 1 p.m. on the implementation of whole genome sequencing that will directly address some of those questions regarding how to do that really in a clinical setting. So I would encourage those folks that have questions about that to attend that session tomorrow. I would like to thank our speakers, Dr. Marshall, Dr. Tucci, Dr. Ebert. Thank you very much for a great session and very interesting talks. And I'd like to thank the audience for their participation and questions as we went through this session. So thanks everyone, and have a great evening.
Thank you.
Thank you.
Alrighty, baby. Let's pause that. Yeah, guys, what did you think? It's a long session, but I found some little gems in there. Some information that I'm going to do a little summary on, hopefully. I'm going to go back and watch through some of the other ones. But yeah, guys, it's been a long day, baby. We done 2 live streams today and 2 videos. So yeah, what did you guys think? Did you get anything tasty fresh? Anything that sparks your curiosity and interest? Thank you, guys. Thank you so much for the donations, by the way. I appreciate it. Man, like Dio and the Mexican Cowboy, I appreciate you guys, man. Thank you so much. Yeah, little bit of the tidbits. I said titty bitties. Yeah, definitely. And we need to have a session definitely on the full broadcasts. I'm gonna put everything together. I've still been trying to collect my thoughts. Thank you, guys. Thank you for attending, man. Thank you to everybody that helped me get into this, because you guys sent donations and it helped me afford it. So thank you so much to everybody that's, a channel member, everybody that is a Patreon as well. It really helps me get into stuff like this, and everybody who's sent me donations offline and on YouTube. But yeah, got some interesting videos to do, some very interesting ones. Inside these actual events, there are literally so many different sessions, and there's so many speakers. There's hundreds of speakers in this ACMG one. So it's definitely must be some sort of world-renowned event. So yeah, it's really, really interesting for me to go through some of the data there and information. And I'm just trying to figure out what is the general movement in genomics going to be, and where does Bionano Genomics stand in that full movement. Yeah, definitely. Did you see? I don't know if you guys saw me, but or if the screen moved, but I was scrolling up just to read some of the questions, and I wanted to see what people were saying. There was a few guys there who were impressed. These guys, thank you, Dom. Yeah, let me know, man. Let me know, brother. Yeah, let me know. Thank you, Katya of Denmark. Thank you, Katya. And Caramel Swirl, Emma Glennie Rat, and Brandon Smith. Oh yeah, baby, you're welcome, man. Anytime, baby, anytime. Thank you, Scoop. I appreciate all of you guys. Yeah, this is what I need to know as well. I'm trying to find basically how are they gonna get mass adoption, is what I'm thinking. What are the stages? So I want to piece together a piece of content for that. And I'm thinking, when I saw the statement put out on a press release about optical genome mapping trying to be written into the guidelines in Africa, I'm wondering what will it take for optical genome mapping to be written into the guidelines over here? Who do they need to impress, to move the NHS or to move Genomics England? Yeah, some people are afraid about selling to China and stuff like that, thinking that someone could crop up with there. And I was looking into the history of who was doing optical genome mapping, and I saw that a competitor, there was 3 companies that were doing optical genome mapping. There was Nabsys, as mentioned in the genomic toolkit, and us, Bionano Genomics. But there was another company, and that company was bought by a Chinese company called BGI. So, yeah, it's definitely something I want to look into as well. It's really interesting. That's a good point. Barriers and patents. Yeah, thank you, guys. But, oh, yeah. It's been juicy, guys. is this their only products? They have the Iris. It was the Iris before. I think there was for Saphyr is the predecessor. Is that the right words? Saphyr was the upgrade. Is that the upgrade from Iris? I heard. But yeah, I need to look into Iris again. I remember watching it on Dio's channel. I was watching it on Dio's channel. Yeah, this one. I think they still sell consumables for Iris as well. So for the people that adopted that machine, I think I was looking at plant genome and stuff like that. There was a lot of plant and animals being run on that system. Yeah, here we go. Exactly, baby. When you see that nano nozzle, oh, he's never gonna show it, baby. Only when the time is right. Yeah, there was who is it running? Is it Nabsys that does the HD? I think it's called HD mapping, their technique. But yeah, that's a good place for me to explore. I want to look at all the competitors that are also in optical genome mapping or similar to the fields. Yeah, when this guy said it, oh, Greg, when the guy said it, you know what? When I had a talk with somebody who works in genomics, and this person said to me that they think, you know, Bionano Genomics is good for research, but clinical uses aren't there yet, because they said, although you can gather all of this data, and they believe it will become the gold standard in cytogenomics, they believe that although we gather all this data and we can find structural variations and we can find out, you know, predispositions for disease, this and that, they're saying that the whole industry is lagging in terms of genetic editing, because genetic editing needs to catch up with what we're currently finding out as well. So they're saying that will kind of slow clinical adoption or something like that. That's what they believed. But yeah, I would love to dig deep dive into it, because this guy we just saw said, you know what, it's ready clinically now. It's ready. So he said it's been ready yesterday for research, but clinically we're ready. So yeah, there's so much to explore. It's fascinating seeing that the world's moving in this kind of way. Think just like decades ago, what was it previously like in terms of diagnosis and stuff like that? Now, if it gets built into the guidelines for all cancer, imagine they do a little sequencing and then bam, throw in some optical mapping as well to make sure you find the structural variations like that leukemia guy. Yeah, take a screenshot, baby. Take a screenshot. Guys, I'm going to try and I need to watch through all of these videos and summarize. But I am pooped. Poop, baby. Different. Is that gaming? Oh, different game. Diagnosis is 2 different gaming. It's versus treatment via edits. Sorry, I don't know what you're saying. Diagnosis is 2 different games. Maybe saying 2 different games versus treatment via edits. True. I think that's what you're saying. But yeah, I want to piece together everything, and I also want to go on the full bullcast, the full bull podcast. But yeah, I'm going to shoot off to bed, guys. I'm going to watch through, try and summarize this stuff. We also have a live stream tomorrow. That's 10:30 p.m. I think UK time, 10:30 p.m. That's going to be an optical genome mapping in leukemia. I think that's another ACMG. So tomorrow, another live stream here, guys. But yeah, love you guys. Thank you so much for attending. And I will definitely catch you tomorrow. And full bull, send me all the details. Thank you, guys. Yeah, enjoy. Don't work too hard, baby. Don't work too hard. You got to enjoy life too. Wishing you all the best, man. Thank you so much. Thank you, man. Yeah, that's what you're saying. Yeah, true. That's true. Appreciate it, bro. I'll catch you later. Mr. Invest a Lot, over and out, baby.